Well, thank you for joining. This is our first innovation workshop of Genesis 2022, and we are joined by some fantastic companies today. We've got Lean Life Science, we've got Earlham Institute and Domainex, and they've come prepared with with some fantastic presentations. So looking forward to this session. Just a few logistics before we get going. If you don't mind keeping your microphones on mute throughout the presentations, that would be great, and cameras off. If you do have any questions, please feel free to put them in the chat box. You can also direct them personally to whoever's speaking, or of course you can follow up after the session via the end. That is it from me. I hope you enjoy these presentations, and over to you, Nicola.
Thanks, Jess. Hi everyone, I'm Nicola Wong. I'm Business Development and Impact Officer at Earlham Institute. So today I'd like to tell you a bit more about our institute and where we're situated. Let me just quickly resume share. Can you see the screen now?
Yeah.
Perfect. So at Earlham Institute, we are based on the Norwich Research Park, which is one of the largest single-site concentrated, research in food, genomics, and health in Europe. So our park hosts 4 different independent, internationally renowned research institutes. So this includes the Domainex, Quadram Institute, Earlham Institute, and the Sainsbury Laboratory. So you can see that on the map there, and we are the red brick building on the bottom left corner there. So we are also joined by other organizations as well, including the University of East Anglia and also the Norwich and Norfolk University hospitals as well. So in the park, we host around 30,000 talented people. This includes 17,000 students, 3,000 researchers and clinicians, as well as 115 companies as well. So we have an outstanding reputation of our researchers, which is internationally recognized. With 10 of our park scientists is in the top 1% of the most highly cited in the world. And the Norwich is also ranked as a top 10 in the UK as a city as well. So a little bit more about Earlham Institute in general. So we are strategically funded by UKRI, which is the UK Research and Innovation. Our vision is to build a future where biology of any organisms can be understood by analyzing its genomes. And our mission is to advance and enable bioscience by applying computational and data-driven approaches. We harness the power of genomics and data-intensive bioscience to answer ambitious questions as well. And we're advancing knowledge through the development and applications of innovative technologies with the ultimate aim of improving plant, animal, and human health. So going on to telling you a bit more about EI's kind of how we structure our organization in general. So at EI, we have both wet lab and dry lab facilities, as well as having a research division, which is the ones showing on the left-hand side. We also offer our facilities and technology platforms as well to the wider science community. So under research, these are the kind of different topics that we are actively doing kind of research and R&D on. This includes data-intensive science. So as a biological science is becoming increasingly data-driven, we aim to lead the applications of data science to life sciences by making data more accessible and meaningful to the end users. And also by developing tools and algorithms, we help to understand it. An example of that is to apply AI and other methods to complex data. So for technology development, by taking a collaborative and multidisciplinary approach to science, we develop novel bespoke approaches to allow us to generate novel data sets, and this will allow us to help answer questions that we were not able to address before. So this could include sequencing the air, single cell, and also using our scientific services as well, such as sequencing and automation, which I'll mention later on. This also includes system and synthetic biology. So by using engineering principles and synthetic biology, we can build and test modules and models to understand network behaviors and genes for regulations as well. And lastly, under research, we also help address global challenges. So these are research linked to human health, including gut microbiome, cancer, etc., but also includes food security and climate change as well, such as crop resilience and agriculture, so that we can help protect the biodiversity in the world around us. So for the facilities and platforms, we have 4 different kind of categories. So under genomes pipeline, this includes advanced sequencing, bioinformatics analysis, as well as single cell genomics as well. So not only we provide it as a service, but we also help with method development as well, if that's something that you're interested in. For BioFoundry, we house suites of state-of-the-art laboratory automation. So together with our expertise in large-scale experimentation and synthetic biology, this can be applied to numerous molecular and microbiological workflows as well. So for research infrastructure, we provide expertise and support for data-intensive research that's used across all life sciences. So this could go from cloud computing to research software engineering as well. And finally, for our advanced training, we provide bioinformatics training from single cell to RNA sequencing and programming, etc., as well as software and data carpentry. So you can tell that's kind of a very wide range of things that we work on, and hopefully there'll be something that will be of interest to you. So coming up next, I'd like to cover a few examples that we've been working on using different technologies and working with different industry partners to demonstrate Earlham's innovation and impact. So firstly, this case study is showing how EI is driving the sustainable fashion revolution. So for this case, the challenge is that textile dyeing is one of the most polluted industry processes. And this caused a severe problem in terms of environment and also potential health issues as well. So there is a need for sustainable dyes that are being developed to perform up to the industry standards without any increase in cost. So for our customer here, it's Colorifics, and they are a rapidly growing SME that is based on the Norwich Research Park. Their patented technology combines the dye production with applications of natural occurring pigments on clothes, and it completely eliminates any solvent use and significantly reduce water and energy requirements. So for this, Colorifics partnered with Earlham Institute with our Genomics Pipelines group and our Earlham BioFoundry to deliver a pathway engineering for optimizing the dye production at scale in microbial strains. So at Earlham, we brought expertise in microfermentation, as well as high throughput screening and advanced RNA sequencing technologies as well. And jointly, the team were able to demonstrate significant improvement in color production in safe industrial bacteria. For the impact, this project represents a critical step in the optimization and scaling of the solution that Colorifics has to offer, and as it is the first truly sustainable alternative to conventional dyeing techniques. So this technology can both use to reduce the pressure on existing water stocks and also decrease pollution, which leads to better well-being and health in textile manufacturing regions. This also opened up lots of different opportunities in the new markets as well for the company, including markets in luxury appeal, sustainable high fashion as well, and new translational opportunities to manufacture antiviral and antimicrobial dyes. So moving on to another example about diagnosing infections in preterm babies. So as you may already know, that preterm babies pose increased risk of life-threatening infections as they are born with underdeveloped gut physiology, immunity, and have an altered gut microbiota. So with these high risks, speed is absolutely critical to be able to detect and diagnose an infection to make sure we reduce the harm as much as we can. So with the MinION technology, as well as Earlham's own analysis pipeline that we developed in-house specifically for this work, the researcher were able to went from the fecal sample collection to detecting the pathogen and antimicrobial resistant profile in just under 5 hours. This is massively reducing, the time it takes to detect this, as the current existing method takes 2 days. So this shows that, not only this technology could save many babies from receiving course of antibiotics that they didn't need, this also helped those who need the treatment most to get it sooner as well, all while tackling the emergence of AMR. Let's move on to another example, again, showing how EI is supporting a new project launch from the biotech industry. So as you may know, Cytiva, formerly known as GE Healthcare, they have developed a device to disaggregate human and animal solid tumor and tissues into viable single cells. So before the launch of the product, they really need to first generate some data to compare the new device with other methods that are already available on the market. So for this, they come to EI and ask us to deliver a contract research project for them, So EI pulled together lots of different expertise from our groups, including the single cell, genomics pipeline, and also our core bioinformatics team. So for this, we managed to deliver different analysis, including single cell transcriptomics, detailed bioinformatics analysis, as well as sample processing and sequencing with comprehensive data quality control, visualization, analysis as well. So the data generated by EI helped enable Cytiva to evidence that comparing to the manual methods, that the device generates single cell suspension with more viable and less stressed cells. So this enables Cytiva to produce an application note for the launch of the new product against a tight deadline. And last but not least, this is a very exciting project that we'd like to tell you about, how EI helped investigate new schizophrenia treatments. So this is a project that is funded by the International Psychiatry Consortium, which is managed by the Medicines Discovery Catapult. So I'll give you some context. Schizophrenia is a neuropsychiatric disorder that is estimated to affect nearly 1% of the global population. Yet we desperately need to have new treatments for this, because current drugs can cause side effects and do not work for everyone. Not to mention that it doesn't improve all the symptoms. So for this, we are working with collaborators from University of Oxford, Biogen, and Boehringer as well in this £4 million collaboration to improve the understanding of the changes in the brain that underlie the schizophrenia for the development of new drugs. And here at EI, we are leading on the bioinformatics analysis of the publicly available data for this acute mental condition, as well as sequencing of the protein data that is generated by University of Oxford as well. By using this newly designed approach developed at EI, we are able to assess which protein isoforms are selectively produced by the target gene in human brain compared to what is expressed in other human tissues, so that we can showcase its specificity and help identify the most promising drug targets for the potential treatments of schizophrenia. So this is a truly innovative and multidisciplinary collaboration that will help enable better understanding of the dominant genes that are present in this complex and underrated disorder, and hopefully will be able to lead to the advance of novel treatments as well. So before I finish, I'd like to tell you a little bit more about how you could potentially engage with us. So we'll work with different organizations and partners to translate our knowledge and expertise and provide access to our facilities and platforms as well, as I mentioned earlier. And hopefully would like to, in the aim to realize the impact of the public investment that Earlham Institute has been given by the government. So this includes research collaboration. We also do employee exchange as well. So there is kind of like annual funded programs where we can have exchange of employees, whether people are coming in or our researchers are going into your companies to have knowledge exchange. This also includes student sponsorship as well as strategic partnership if there's something that will align with our strategic interest between your company and ourselves. This also includes contract research and consultancy as well as scientific training as well. So this is just a list of a few more other companies that we engage with. If you want to find out more, please let us know. So this is my details here. Feel free to contact me via email below if you want to find out more or want to explore any ideas going forward as well. I'll also be attending Genesis in London as well. So I hope to see you there. Thank you.
Brilliant. Thank you so much, Nicola. So up next we have Ray from Domainex. So I'm just going to bring you up, Ray.
Perfect, and I'll just start sharing my screen.
Perfect.
Can you all see that?
Yeah, that looks good.
Perfect. All right, well, nice to meet everyone, and thanks for joining this session. So I'm going to tell you a little bit about Domainex. I'm the Exec VP of Medicines Research here. So for those of you who are not aware of Domainex, we've been around for 20 years, and we're an established integrated drug discovery CRO based in the Cambridge area. However, we have global reach. We work with biotech and pharma across the globe, as well as a number of academic institutions, really enabling their drug discovery process all the way from gene to candidate. And here is just a selection of some of the people that we've worked with and collaborated over the years. So during that period, we've really brought together a world-class team of over 90 scientists currently, but will be getting over 120 by early January. Majority of those scientists across the disciplines, which I'll come on to in a bit, are pretty much 70% have PhDs and postdocs, with about an average experience in the drug discovery sector of around 8 years. And through this breadth of experience and that we've brought in, we've been able to deliver a number of clinical candidates for our clients, as well as have a number in preclinical development that's currently going through that pipeline. But we've also added value to a lot of our clients by generating novel IP, and we're inventors, or the scientists in the teams are inventors on over 150 patents and numerous scientific articles as well. So we've brought together a multidisciplinary team. So this goes all the way from chemistry, assay biology, and protein science. Now, we originally started as a protein science company, and we really like to try and utilize that in order to structurally enable our projects, but we can work in every single paradigm as well. So within the chemistry teams, we have really experienced medicinal chemists, synthetic chemists, analytical bioanalytical teams, as well as computational teams, enabling that small molecule research primarily, all the way through to lead optimization and candidate selection, and working hand in hand with the assay biology team to create assays in order to set up screening, but also to really understand the physiology of the compounds. And we're also renowned for our biophysics platform that we've heavily invested in. So companies come to us in order for us to work on their programs all the way from gene to candidate selection in a really integrated fashion. However, a number of our clients we also work with aren't ready for that approach, or have got specific issues that they're tackling on their current projects internally. And we support them to do that with a number of different platforms that we use in an integrated fashion, but we can also use to unlock challenges that everybody is currently facing. So going from sort of the gene area, as I say, we're highly experienced in protein expression, whether it be in extracellular proteins, but also unlocking more challenging membrane proteins, and as well as in the structural biology, obtaining soluble protein in order to deliver crystal, in order to get crystal structures, and more recently Cryo-EM, and I'll come on to that in a little while. We also use screening teams in order to obtain novel compounds, in order to unlock targets, in order to get new small molecules. So we can do virtual screening, fragment screening, as well as covalent inhibitor screening as well. And this also goes alongside high throughput screening as well, which we can use to unlock different projects. And this is then really underpinned by the assay biology, making sure we've got key screening assays in place, but also looking at screening cascades and making sure we're looking at selectivity and various other paradigms as we move to sort of hit-to-lead optimization of those compounds, where we bring in those experienced chemistry teams and medchem teams in order to make sure we're optimizing compounds. And again, this is all underpinned by the other disciplines, including ADME and pharmacology. But the really emphasis here is that there's no one solution that fits everybody. So we're highly collaborative and work to customize our science in order to make sure it fits in order to unlock your targets of interest. So once we've got small molecule hits, whether it's from our internal platforms or from our clients, we then look to take this through all the way through the drug discovery process. And we're with you every step of the way whilst we do that. We've got experienced people who have worked in the industry for over 30 years, where we're looking at establishing the target product profiles of those small molecule compounds, and then continually reviewing them to make sure that they are fit for purpose as we go through the more expensive end of animal model studies, and also scale up in order to go to IND ready status. And in order to do that, we work with a number of strategic partners who we've worked with for over a decade, and to name a couple here, Crescit and Pharmadex, which again, both local to the Cambridge area. So once we've got those hits and they're validated, we really encourage and really express with all our scientists in the med chem area that it's not just about optimizing one factor when it comes to, for instance, potency of compounds. And we really look at the analogy of the Rubik's cube, where everybody can solve generally one face of that cube, be it in this case potency. But in order to develop and optimize a compound of interest, there are a number of different parameters which you must understand, and each of them impact the other. So in order to solve that Rubik's cube, you've got to understand and make sure you're keeping an eye on all of those different criteria. And we really work in a multi-parametric optimization fashion, where we emphasize that every compound that we design is important and work on the basis of every compound counts. And this then really has enabled us to generate high quality compounds for our clients. But it's also about speed and efficiency, and it's going around that design, make, test cascade as quick and as efficient as possible. And we've built up our teams in order to give them all the required software and computational tools and experience they need to do the design, go through to the synthesis of those compounds and generate those compounds in as quick as possible, to go through to screening, when we've delivered data in an average 5-day turnaround time, depending on the assay, and whether that's in biological output or also in the ADME, in order to get that data, to do the analysis. And again, we have all the databases and facilities in order to make sure we can generate and do that analysis. And for us, as I said, it's all about collaboration, making sure that we're talking through these results, making sure our clients are understanding them, and working in collaboration to generate the best compounds as possible. And we go through that design, make, test paradigm roughly about 5 times in a 3-month process. And so we can make all those key decisions hand in hand with our clients. And as I say, then we've worked in this in a number of areas. We've got a proven track record of success, but we've also invested in a number of different solutions that are enabling timely and cost-effective solutions for our clients. Our teams are highly scientific driven, and going on to that customizable nature, everything is tailored for you and your projects. And again, we've invested in a range of technologies in order to make that happen. So a couple of areas that are obviously in vogue, I just want to highlight them at the moment. So protein degradation. So we've worked in the PROTAC drug discovery area for a number of years. We've taken compounds that we've prepared up to multiple gram quantities, enabling PK and animal work. So we've got all the experience to do this. We've worked on known E3 ligases, but also worked in targeting novel E3 ligases that our clients have identified, or again, identifying through virtual screening or fragment screening, novel ligands in order to target the current E3 ligases. And we've set up a whole platform in order to understand the degradation of proteins, but also understand the binary and ternary complex formation of those compounds. And again, bespoke to Domainex, that innovation created cell accumulation assays where we can look at how much of those PROTACs have got into the cells of interest, again, so we can optimize the correct chemotype and make sure we're working on the right compounds going forward. And then mentioned earlier in the Cryo-EM space, with an established protein science company of origin, we always generate high quality protein, one of the key factors, and we've also been able to attract a number of key structural biologists with Cryo-EM experience. We're based in Cambridge. We have access to the state-of-the-art facilities with a number of microscopes, and through that, we're now generating Cryo-EM maps in order to unlock targets and work on structurally based drug discovery programs that we can start to interrogate ligands in order to pass that through that process to generate those clinical candidates. So it just needs me to start wrapping up. So we've put together a talented, experienced, multidisciplinary teams. We've got cutting edge technologies to unlock those challenging proteins, and we work in a highly collaborative manner. This has enabled us to generate high quality, cost effective and timely data driven projects that's really customizable to your needs, generating value by unlocking novel IP and taking your compounds all the way through to the clinic. And then I'll just finish off with a slide just so you can have a look in your leisure relating to the website. There's various links there, but I'll just highlight a couple of press releases we've had more recently where we're working with Parkinson's and NRG in the neuroinflammation space for Parkinson's research, but also in the oncology area where we've unlocked a challenging, unthought of, undruggable target protein-protein interaction of the FLIP inhibitor program, where it enabled Queen's University Belfast to go through a licensing agreement with Ipsen. And so, yeah, if you have any questions, either reach out to me or I'm more than happy to answer any now. So, and thank you for all your attention, but also for the Domainex team in order to be able to put all this in place in order for us to work in that drug discovery area.
Amazing, thank you, Ray. So that leads us nicely on to, last but by no means least, Lean Life Science with Ned. So I'm just going to bring you up, Ned. Brilliant. You're just on mute at the moment.
Okay, can I show my slides? Is that okay?
Yeah, I can see your slides.
Okay, perfect. Hello, everybody, and thank you for the opportunity to present today. So Lean Life Science was literally just launched last week, and it was sort of developed out of a need that I saw in my last job. And it's an approach, it's called Lean Life Science because it's an approach to developing life science assets. And this program that we just launched, Oncology Development 2, is in partnership and funded by Cancer Research UK. The whole focus of Lean Life Science is to catalyze innovation, if I can drop this, in the life science industry. And it's focused on leveraging the unique collaboration of expertise spanning the R&D ecosystem. It uses our innovation development framework to provide critical insights, validated delivery plans needed to accelerate the translational journey of life science innovations to highly desirable, de-risked, investable opportunities. So I was trained as a scientist. I worked for 12 years as an investment banker in London. My job was to help life science companies raise funding, and we did everything from identifying them to due diligence to introducing to investors and running the whole investment process. And in that, I often saw a challenge in that what we primarily did was package what companies had and help them, teach them how to tell their story to investors. But what we didn't do is there are a lot of gaps often in the investor stories, and we didn't really help them fill those gaps. And except for maybe a few introductions to some board members or management that we may have had in our network. I then ran I was hired, well, my company was hired, but I led the incubator inside AstraZeneca at Alderley Park. And when it was sold to Manchester Science Partnerships and Bromwood SciTech, my job was to really build the ecosystem of support there. And so I built the incubator programs, accelerator programs, working with the director, the national faculty director of the National Science Foundation in the US for I-Corps, with the Scale-Up Institute and Babson College for scale-up programs. I set up a mentor network of over 240 people, led on the forming of a venture fund that our company managed and all of the sort of workshops. And out of that, we had 86 spinouts from AstraZeneca. There are now over 200 companies, 3,500 people. And I felt really kind of fortunate to be able to work with so many bright and talented people. But I also, in doing that, recognized the challenges of our own programs, and that we'd often run these 3-month programs and people would say, that's great, you've told us what good looks like and you've told us what we're supposed to do, but you really haven't helped us put in place our delivery plans and delivery partners and really to the next steps of support. It's more, and I sometimes had people almost get angry at me. But nevertheless, they were really effective in that when we looked at the companies we supported versus those that we don't, they grew 2 times as fast, they raised 2.5 times as much money, they were 3 times less likely to fail. But I also recognized the challenges and heard from a lot of the stakeholders. So pharma was saying, you know, we used to develop all of this, or AstraZeneca was saying inside AstraZeneca, now we're more porous and we're looking to, you know, license projects in, but they don't come pharma ready in the way that we develop them. Tech transfer offices were good at protecting their technology, but less good at really translating it and commercializing it. Charities, you know, cancer research is a good example, is, you know, spend a lot of money on funding their science, and now really want to get that science funding out to the benefit of patients. And so there's more and more of a focus on translation and commercialization. Government funding body like Innovate UK has often funds these kind of value of death or risk companies sort of with this market need or market risk, and I think haven't been as successful in, or they haven't had the rate of success of commercializing those as possible. And I think part of it again is sort of that gap in missing the really how do you develop the assets. Hospitals looking for new technologies, national translational centers and contract research organizations, often really useful at benefiting translational projects, but sometimes those projects aren't funded, and partly because they don't have an investor story. And SMEs, really fantastic science, but often not having the expertise that they need, and sometimes not knowing where to find it. And so the idea of what we set up really was to pull this all together in a collaboration with expertise spanning the entire ecosystem and through partnership. So we set up a partnership with across this ecosystem and aligning the interest. The first program that I ran in my last job was called Oncology Development Program, ODP1. We had 8 partners, and the final 8 companies raised £40 million. So part of what we did is we really helped with this sort of, we started off by doing a national call. We had pharma partners and some of the partners on the program help select the projects. So you had your end buyers or potential exit partners selecting the projects, providing feedback for the data that they need to see in order for them, you know, really the next kind of data points or go-no decision points. We use the program to provide this framework for line of sight. And then we used experts and CROs to really provide the project plans and delivery plans. So the companies ended up with a pitch deck that had this use of funds, value milestones, a validated line of sight, expert partners potential exit partners saying we're interested in these and providing data on what they wanted to see, and then delivery partners, such as CROs and professional services firms, providing here's the path to, or here's the use of funds, the project plan, and the value milestone. So all the stuff that when I was raising money previously as an investment banker, oftentimes they were sort of the missing 4 or 5 slides. And again, the proof was in the pudding, in that those final partners, the final companies raised over 40 million. In the last program, and the first program I ran in my last job, we had 8 partners. Every single partner that I've spoken to, there's 2 pharma companies now, but we've got actually 2 more confirmed, 2 more in discussions, and another 4 that we're also talking to. We've got 20 CROs signed up already, and a lot of this is just about everybody that we've talked to have said, this is a model that we can see provides a lot of value. We're part of this ecosystem. Developing assets in our ecosystem really requires this collaborative approach, which actually you've heard in the previous presentations. And this is we did a survey of the partners, and this is the areas of expertise that they cover: therapeutics, diagnostic biomarkers, platform technologies, digital computation, and oncology. The framework is one that is fairly ubiquitous now. So iCure uses it, iCore in the States. A lot of the accelerator programs use it. It's been written up in Harvard Business Review and in Nature. The National Science Foundation, National Institute of Health use it. And it's a fantastic tool. And so we've trained with the people who have developed the tools, Alex Osterwalder in Strategizer. But the tool, you need to be good at tool expertise, but you also need domain and commercial expertise. And that was the point of us really bringing in the partners. And so you use the tool to validate your line of sight, but then you use your partners to really provide the expertise that's needed and the project planning that's needed once you've validated where you're going. And you use them also to validate where you're going. So the program that we're doing is the first in series of programs. How it's going to work is we'll have day one is what good looks like. And so very low barrier to entry. Almost most people that are interested, we'll have pharma in licensing, BD, search and evaluation people saying, this is what good looks like. This is how you should approach us. Investors, similar sort of thing. A case study saying this is the value that the program's offered to me. Day two is, but part two is really a 3-day program, which is more of, talks on TPP, 5 Rs, safety strategy, regulatory strategy, IP strategy. Day two on an introduction to the framework and how it's applied in our program. And day three is an opportunity to engage with the partners and the experts. We've also got 30 oncology experts in our program. And then the third part of the program is really doing the deep dive gap analysis, validating the line of sight, exploring your commercial and business strategy, putting in place the project plans, the business model, and really building that up so that you end up with this kind of validated pitch deck, and then introducing you to investors. And last time we introduced them to 44 investors. There's a very old picture of me. I've mentioned my background. One of the differences in this program is, as I mentioned, you need domain expertise. The people that we have, Steve McConchy is a clinical trial expert. He just sold his company Aptis Clinical to Catalyst, so very successful, knows about clinical trials. Nigel Brooks is an ex-project lead in oncology in AstraZeneca, director of the Manchester Cancer Institute, and then head of translational science at CellCentric. We've got someone who's used the tools inside Big Pharma, to asking her teams to present their projects within the scope, within the tool themselves. And so one of the critical aspects is that you understand the tools within the context of the industry that you have, and you've got people around you, your experts, who can not only teach it, but also give you that kind of that market and domain expertise on what the expectations are and what you need to be seen. Lynn is ex-Novartis, runs her own company now in communications, and Claire is absolutely invaluable. She's our executive assistant. There's the website, Twitter, and an info @ and you're welcome to contact me directly, which is either through my LinkedIn or my email if you're interested in partnering with us, being a potential investor, or being introduced to some of the companies on the program, or applying to the program and learning a bit more about it. We're going to run some some webinars so that people can come and take an hour to kind of listen in more detail on what we're going to provide during the program, ask questions. And then day one again is really more informative for people helping understand what the kind of exit partners and investors are interested in from our industry. That's it. Thank you.
Thank you, Ned. So we have a little bit of time for questions. If anybody has any, please feel free to put them in the chat box. Thank you, Nicola, Ned, and Ray, for your fantastic presentations. We will give it a few moments and see if anyone's got any questions. Otherwise, please do feel free to follow up with our speakers after the session, and get in touch. I can't see any in the chat box. Oh, any questions from you, Alan? Let's see. Oh, it disappeared off the camera.
Thank you. I'm just letting you know I am listening, but no, I haven't anything specific. Thank you.
No worries, that's fine. Nice to see you as well. All right, wonderful. Well, we'll wrap it up there. Please do join us for the next session, which starts at 3 p.m. And that one is ERDF CPI Sprint, and that is technologies to accelerate the development of novel therapies.
Very good.
And we've got another innovation showcase tomorrow, and that is with Aviade Bio, Imagine AB, Eagle Genomics, and the Quadrum Institute. So we look forward to seeing you all there. Thank you for coming.
Thank you very much.
Thank you.
Thanks very much, all.
Thank
Bye.
you.